We'll begin first with a look at why we have organelles in the first place. How do they come to be inside cells? Organelles, just like organisms, so just like we have organisms which are made up of many cells, right? So we have an organism. Now an organism is made up of many organ systems. Okay, so an organism is made up of many organ systems, and they have their collective functions, or individual functions, we should say. Okay, so for example, an organism needs the digestive system because that is the way the organism obtains biological molecules that it can use to synthesize its own biological molecules. So it acquires biomass, and then it breaks those molecules down so it can build its own molecules. Okay, it will have transport systems. So transport systems allow the organism to distribute substances around the body, allowing communication, allowing supply of oxygen, waste removal, things like that. It might also have communication systems, such as the nervous system, endocrine system, that allow coordination between the distinct, different, and distant parts of the body to allow coordinated actions. So, just like in the organism, in order to survive, needs all its different organ systems. Each of these organ systems is made up of tissues, organs, individual organs that have more specific functions. Okay, so each organ, each system is made up of its different organs, and those organs all have individual functions. Okay, for example, in a digestive system, the stomach's role is to churn the food up, mix it with digestive enzymes, create the conditions for those proteolytic enzymes to work optimally. And the large intestine, for example, its main role is not digestion per se, but it's more to absorb water, any remaining water from the food. And similarly, the mouth's job is to carry out the physical digestion of the food, mix it with those enzymes in the mouth. So you see how all those organs have their individual thing to do that contributes to the organ system. Similarly, each of these organs, in order for them to carry out their function, each organ is made up perhaps of a collection of tissues. Alright, now each of these tissues is highly specialized, formed from similar cells doing similar things. Okay, so you have these tissues, and each of these tissues is formed from cells. Now, until this point in our discussion, cells are the basic unit, but as we'll see, that cells are not really the basic unit anymore, because cells themselves are made up of things. Okay, so each of these tissues is made up of collections of cells. So each tissue, as the definition goes, made up of cells similar in structure and function, doing a similar collective job. Okay, so you have all these cells doing these things, forming tissues. However, even for the cell to function, even for the individual cells to function, it needs different bits and bobs. Okay, so for the cell to function, and do the job collectively with other cells in the tissue, each individual cell itself has its own components doing different things in the cell, so that if you put all those functions together, it allows the cell to do its job. If the cell then does its job, the tissue can do its job. Okay, if the tissue does its job, the organs can do their job. Collectively, the organs doing their job means that the organ systems are working, and if the organ systems are all functioning properly, then that means that the individual organism can function properly, survive. Okay, and maybe we can even extend that to populations. Okay, so if collectively we all do the right thing, then the population, the species survives, and if species survives, then ecosystems survive, and if the ecosystem survive, then the whole planet continues. Okay, so it all comes down really, as we all knew, to biology. Okay, so let's now discuss how these organelles came to be in the first place. Okay, so let's begin in the ocean again, and let's start with a very basic form of life. We're going to start with a prokaryotic cell. Any living organism, one can argue, is only there to serve the purpose of reproducing its genetic material. So any living organism is going to have some form of genetic material. Okay, so we are going to be looking at bacteria. So they have that circular DNA. Okay, that circular DNA has to have a means by which it can reproduce itself. So it needs some enzymes, it needs some ribosomes that will be able to translate the mRNA that's being produced from the genetic material. Okay, and in order for this organism to be able to have some control over its environment and to be able to keep these valuable structures to itself, it needs to have some form of boundary. And that boundary is the cell membrane, or the cell surface membrane. Okay, so by having this membrane around it, it can keep the contents that are important for its survival inside. It can keep the concentration of these things high enough so that there's a reasonably good chance of a collision between the two things that you want to collide, namely enzymes and substrates. Okay, and you contain your genetic material so that the cell is able to reproduce it and produce a copy of all the components that it needs. Okay, and then the prokaryotic cell would have a cell wall around it, perhaps as a means of attachment to things, perhaps as a means of protection. Okay, and then you might also have a flagella to make sure that the cell can move towards areas where it's got a higher chance of survival. Okay, so this is our very basic prokaryotic cell. Okay, now even the prokaryotic cell, as we can see, has got unique structures that allow it to survive in this environment. But it's got specific structures that do specific things. Okay, now in a prokaryotic cell, the cell membrane is very important because the cell membrane is where many of the critical functions happen. For example, forms of respiration will be happening on the surface membrane, right? So on the surface membrane you'll have the prokaryote version of respiration going on. Okay, so any electron transport and ATP synthesis, that will be happening on that membrane, whereas for us it happens in mitochondrial membranes. Any photosynthesis might be happening on such membranes. Okay, synthesis of the lipids that contribute to the membranes and make up its structure. Lipid synthesis will be happening here. So all the enzymes and proteins that are required for these things to happen are located on the prokaryotic cell membrane, and that's where all these unique and differentiated functions are happening. But here's the thing, guys. Here is the thing, that prokaryotes are actually very small. Okay, prokaryotes are very small compared to eukaryotes. So eukaryotic cells are very large. Right? So if we have a eukaryotic cell, it might be something like that size compared to a prokaryotic cell. Now, the prokaryotic to eukaryotic evolution was quite important because, relatively speaking, the smaller you are, the more energetic requirements you have. The bigger you are, relative to your size, the fewer energetic requirements you have. And so it was probably a very important evolutionary advantage for cells to be bigger in terms of volume. They probably could store more food for themselves, more energy storage. They could probably maintain temperature conditions much better. And so they had an increased chance of survival from that point of view. However, there was one big problem. And that problem was that as you go from a prokaryotic cell, so a prokaryotic cell has, relatively speaking, it has a high surface area. It has a high surface area compared to its volume. Okay, so it's got a high surface area to volume ratio, which means all those functions that it was able to do on its cell surface membrane, there was plenty of membrane, enough membrane for it to do all those functions: photosynthesis, respiration, lipid synthesis, all those things. There was plenty of cell surface membrane for the size of the cell. Now let's cut to this eukaryotic cell. This eukaryotic cell is quite large, but according to its volume, so now its volume is quite high. Okay, but as we know, the larger objects get, in relation to their volume, the surface area becomes smaller. Well, at least the ratio becomes smaller. So even though the overall area of the membrane is increasing, the amount of membrane that you have compared to the volume becomes smaller. So this eukaryotic cell, we could argue, simply does not have enough membrane in order to carry out all the functions that this cell might need it to do. Okay, so one of the things that's been proposed, and this does get supported by what we see in the cell, one of the things that is proposed is that what started to happen was that inside these cells, what started to happen was that the membrane started to fold inwards like so, and by doing so, by the membrane folding inwards, it was able to increase its surface area adequately enough that now, by all this extra infolding of the membrane, it was now able to provide enough membrane to allow all the functions that this cell needed to be carried out. Okay, so this membrane is now folding inwards, and by doing so, there's much greater surface area, and now the functions of the cell, or the surface or the membrane area of this cell, is now adequate for it to survive. So now, in this situation, we have enough membrane inside the cell that all these different jobs that the cells need, now enough lipid can be synthesized, now enough of these other functions can be carried out. Okay, so that's one situation, and one could imagine that at some point they lost the ability to be continuous with the extracellular fluid and became internal organelles in some way. Okay, so they lost their connection to the outside, and now they became fully internal organelles. Okay, like so. Right? So this is one argument for how these organelles came to be. Right? That the membrane inside, or the membrane started to fold inwards in order to satisfy the requirements of the cell, and in doing so created these internal membranous structures which we now call organelles. Alright, so the organelles that come under this category are the nucleus. So the nucleus is that thing right here. So as we saw, we can imagine how it formed. The other thing also, the endoplasmic reticulums, both rough and smooth. Both rough and smooth are considered to have evolved in this way. Additionally, the Golgi apparatus, also similar, and the lysosomes and the vesicles. Okay, so this family of organelles is thought to have formed by the infolding of the membrane. Okay, now two very important characters which we have not talked about yet, which is the chloroplast and the mitochondria. Now these are special cases because the chloroplast and the mitochondria themselves resemble prokaryotic cells. So when we look at the mitochondria, we can see that mitochondria have an outer membrane and they have an inner membrane. They have two membranes. Okay? So and not only that, but they have their own circular DNA. Now that is unlike the other organelles that we have discussed. So it is thought that organelles such as the mitochondria and the chloroplast, they were once independent cells of their own. They were independent entities, independent prokaryotic free-living organisms, but then what happened was that they started to form, or there was some advantage for them to be in association with other cells. So maybe they needed materials that they were getting from other cells by living in proximity to other cells. Maybe these other cells were getting some benefit from being in a close association. So there was a symbiotic relationship between these cells at some point. And it would then follow that perhaps these cells, with their own DNA inside, with their own cell membrane, perhaps these guys started to get internalized. Okay, so they started to get internalized into the larger, or the cell with which they had a symbiotic relationship. So there was another infolding of the membrane, so just like an endocytic process. Okay, so the membrane of the original eukaryotic cell started to invaginate the prokaryotic free-living organism, started to get internalized, and then we can see now the beginnings of an inner and outer membrane. So if we follow this process through, what would happen, if we follow this process through, this cell eventually goes completely inside. Okay, so once it's inside, I will tidy this up. Once it's inside, so what happens then is that this cell is now inside. Move that arrow. Okay, the membrane that internalized around it remains around it. Okay, because once it gets inside, it kind of pinches off. And now we have an organelle. We have a structure inside the cell that has its own original membrane that belonged to it. That's the inside membrane. That's the internal membrane. Okay, then it also has an outer membrane that has formed from essentially the endocytic process or endocytosis. So that's its outer membrane. Okay, so and if this is true, they should also have their own original genomic information with them. And as we see with chloroplasts and with mitochondria that this is the case. So some organelles have formed as a result of endocytosis and symbiosis. We're going to combine those as endosymbiotic. So these organelles are the chloroplast and the mitochondria. They both have internal and external membranes, i.e. envelopes. They both have genomic information of their own as well. Now, as it stands today, those genomes are not completely dependent only on those genomes. So some of the mitochondrial and chloroplast proteins that are important to them are actually made in the nucleus. So that symbiotic relationship has become more close over time. Okay, so now these organelles are very dependent on genes from the nucleus. They are not only dependent on their own genome. So, ladies and gents, there's one more reason why it is very important for cells to have organelles. Okay, and that reason is the most common reason that is given, which is that when you have structures that are bound by their own membranes, you can control the conditions inside those organelles to be optimal for the functioning of that organelle. So once you create, for example, once you have an organelle that is smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is bound by a membrane, then all the enzymes that the smooth endoplasmic reticulum needs can be contained within the structures of the smooth endoplasmic reticulum, bound by membranes that can control movement. Okay, so that's a very important feature, that membranes act as barriers. And because membranes act as barriers, we can control movement across the barrier. Movement. So membranes act as barriers. Barriers can be used to control movement, and if we can control movement, we can control the contents. And if we can control the contents, we can control the conditions, and we can control the function. So if we're only allowing certain enzymes into the smooth endoplasmic reticulum, that means that we are essentially saying that the smooth endoplasmic reticulum is going to have a certain set of functions determined by the protein content that it has. Okay? So that's really important, and if we can do that for the smooth endoplasmic reticulum, we can do that for the rough endoplasmic reticulum, right? We can make sure that only certain proteins remain in the rough endoplasmic reticulum, and therefore the rough endoplasmic reticulum is going to have different proteins. The rough endoplasmic reticulum is going to have different proteins within its membranes than the smooth endoplasmic reticulum has within its membranes, and thereby we can start to assign them different specific jobs to do because they have different proteins that are doing it. Okay, so that is a very important feature, because once you can do that, then all these structures, such as Golgi, they have all these different functions, but the reason why they have these different functions is because their protein content and their structure is very highly controlled, because they all exist within this slightly different membrane controlled environment inside. Okay, so membrane barriers allow compartmentalization. Compartmentalization. That's the third reason. Right? Okay, so just to recap then, so let's do a quick recap about all this stuff. Okay, why do they exist? Because each organelle should have a specific function and can be specialized. Okay, and because each organelle can be specialized and have a specific function, it contributes to the overall function of the cell. So that's reason number one. So the second thing was that as eukaryotic cells are larger, they need more internal membranes. They need more internal membranes in order to do all those different roles. So those functions need to start getting internalized in order to have enough membrane to carry out all those functions. Okay? And lastly, what we discussed was that if you have different compartments, compartmentalization, if you have these different compartments, you can control the conditions, and then each of these different organelles can work. So they can have unique structures, they can have a unique and specialized conditions, and because of that, they can have unique functions. So in some way, all of these are interconnected, okay? But this is very important for us to understand before we move on, because there's a lot of stuff going on inside cells, and one of the things that might make it easier and I really do believe this, is that one of the things that makes biology easier is to understand the bigger picture and the deeper reasons for the way things are. And if we understand those things, then the factual knowledge makes more sense. Okay? If we understand how the nucleus, the rough endoplasmic reticulum, the Golgi apparatus all work together, then it's easier for us to understand or remember their individual roles. So this was the first in the series on the organelles. We'll now go step by step through all the organelles and have a look at their structure and how that relates to function.